Thank you, Andre, for this really, it's I think the first time I get introduced in this social media savvy way. So I'm going to see how much my follower number increases. It's a real pleasure to be here. It's really an honor. I would like to thank the organizers of this unique event. It's also kind of a bit special for me because although I'm from Switzerland, I am from Portuguese, Spanish ancestry. So I think I'm kind of the genetic embodiment a bit of this meeting. And I'm especially happy actually to chair the session today. So we have been looking and hearing about amazing work being done at the level of care and also research to understand mechanisms in Alzheimer and neurodegenerative diseases. And I think this session this afternoon continues this really theme. And what I really find interesting about the selection of the three speakers is that it will illuminate the question of what are the mechanisms underlying circuit function and then also circuit malfunction from very different angles. So we're going to hear really three world leaders in their topic talking about neurodegeneration and Alzheimer from very different perspectives. And the first speaker is Richard Mayeux. He's the Gertrude Sergievsky Professor of Neurology at Columbia University. He's also the chair of the Department of Neurology and the neurologist in chief at the New York Presbyterian Columbia University Medical Center. He has a long-standing interest in understanding how aging affects the brain and how Alzheimer interacts with aging and the genetic makeup together with environmental factors to understand the mechanisms underlying Alzheimer. And he's really pioneered the use of genetic and epidemiological methods, fuse them together to really understand at the biomarker level how the environment affect and the genetic makeup affects the development of the disease, and then also really move on and try to get at the mechanisms. Thank you very much for being here, and we look forward to your talk.
Thank you. I'm very pleased to be here as well, and I thank the organizers of the conference for the invitation. And I'm also delighted to have the opportunity to explain the Alzheimer's disease sequencing project to you. So Alzheimer's disease, you've seen this slide earlier. There are 50 million people right now that we think have the disease. Those are the ones that we know about. There are still many that we do not know about. They're still living in their houses, well protected. And if the trend continues without some interruption, we can look in 2050, which isn't that far away, to almost 130 million if things are not corrected. The problem is there's no effective treatment. And one of the reasons to do genetics and to explore genetic targets is that there is a hope that we would find a modifiable target in one of the genes that we're looking at. So the Alzheimer's disease sequencing project is really an international effort to investigate the entire genetic basis of late-onset Alzheimer's disease. And it started off with the Alzheimer's Disease Genetic Consortium, which was largely based in New York, and then evolved, in the US, and evolved into the Alzheimer's Disease Sequencing Project. And there are partners for this program. There's the Alzheimer's Disease Coordinating Center, which links all of the 30 Alzheimer's disease research centers in the United States and allows us to sample DNA from all of the samples that these centers see. All of the DNA is held at the National Cell Repository for Alzheimer's disease, such that if you want DNA, if you want tissues, if you want CSF, you can go to the cell repository and request it. It's a simple application. You get the data and you get the information. All of the data that's generated through the Alzheimer's disease genetics projects are stored in the what's called NIAGAS, or the National Institute of Aging Genetics of Alzheimer's Disease storage site. It's frequently updated, and with the fact that dbGaP is no longer going to store or share sequence data, at least for the time being, you can apply to the NIA-GAD to get access to whole exome and whole genome data. And then the last one is the National Institute of Aging late onset family data. We have about 2,000 families worldwide that we followed. That study was just renewed, and we collect data on families that are multiply affected by Alzheimer's disease, and now we're working on the offspring of those families that we acquired over the past several years. The principal investigators are listed below, and our main goal is very simple. We want to try to find some potential targets. So let me get right to where we started, and that was with genome-wide association. The first study that looked on a large scale for Alzheimer's disease was done by Julie Williams' group in England. She uncovered these loci, and I really restrict my comment to loci, because in GWAS, we are never quite sure if the gene that we're looking at is really where the loci is. We hope it is, but we're never quite sure. And that was followed 2 years later by a large effort in the US to put together all of the Alzheimer's Center data plus a number of cohorts, which confirmed many of these sites, but gave us the idea to join forces across the, I think they say across the pond or across the Atlantic Ocean, to put one group together, and that led to something you've heard about earlier today, the International Genome-Wide Association Study, or IGAP, where we had 74,000 people with GWAS data, half cases and half controls, and it pulled out most of the targets that we are all working on, and many of them were mentioned today, some of which have been sequenced, and I'll talk about that in a few minutes. Many have not yet. And the strategy for understanding these loci is that at the loci level, again, you don't know what's driving the association. It could be something in linkage disequilibrium, actually quite far away, or it could actually be something in the gene itself. So the onus is on us to go back to visit these loci to see if the gene really has a mutation or a set of mutations that are accounting for the association. And that's been much of the work that's been underway recently. We've also had the opportunity to put together some minority populations, something that had never been done because the sample sizes were so small. This is about 6,000 African Americans. It's still underpowered. We're going to revisit that this year, but we were able to pull out of this ABCA7, which is almost as strong a risk factor for Alzheimer's disease as is ApoE. ABCA7 locus sits right between the two Caucasian papers, so this region becomes very important, and I'll come back to that a little bit later, and you can see that it's all in one large linkage disequilibrium block. The gene itself is important for transporting molecules across plasma and intracellular membranes. It's involved in HDL, the formation of HDL when it's exogenously expressed, may be involved in phagocytosis related to injury. But the truth is we really don't know what its function is in Alzheimer's disease. This is what it's known to be doing, but we don't exactly know what it does in Alzheimer's disease yet or how it increases risk. Another study based entirely in the Dominican Republic, which is another study site for my program, we identified ubiquitin protein ligase, FBXL7. This has a smaller signal in other cohorts. This is an interesting population because it's 40% admixed of African origin and the rest Spanish, and then a small amount of indigenous population. But again, ABCA7 and ApoE appear to be strong signals. So putting all of the data together from the GWAS signals, two papers recently have sort of illustrated how much of the genome have we accounted for. This was from a study in PLOS Medicine, which took all of these cohorts, put them all together, looked at all the GWAS signals, and can account for at least 10-year differences over ApoE4 alone if you account for the effect of some of these genes. If you want to look at the individual genes, you have to use the family-based analysis, which came out almost at the same time. And overall, we can account for 70% of the increased risk by these loci. And again, they're loci, they're not all genes. Some of them are genes with variants in them. Some of them still remain loci. So what about sequencing? Well, we've started an effort to, when it was relatively inexpensive to do targeted sequencing, and then of course it became cheaper to just go right to whole exome sequencing. But at the beginning we used targeted sequencing to visit some of these targets, these loci. And the most striking thing was our whole concept of early onset versus late onset disease became sort of questioned, because we found several APP, presenilin 1 and presenilin 2 mutations in late onset families who didn't really have a dominant pattern, but they had a consistent familial pattern. And this was in the study by Carlos Chugaga at Washington University, and a similar study in a group of Dominicans by Joe Lee from our group. And the same sets of variants showed up at about the same frequency. So these are mutations that are present in late onset families, not early onset families. We also had previously used a SNP approach to interrogate SORL1. We had identified 3 SNPs that were genome-wide significant, although it wasn't a genome-wide study, in 2 blocks. This included about 6,000 individuals. We subsequently, a couple of years later, sequenced this group and found, sort of went too fast there, we found 3 variants that account for most of the association in 3 distinct regions. And they all have an effect on A-beta processing. Mutations are associated with increased A-beta 40, 42, and soluble APP beta. And the main effect is on binding of the SORL1 protein to the APP, or the A-beta fragment. Just to share more of the sequencing data, we've now tried to sequence all of the loci that are in the GWAS signals, so all 30 of them. And even though the sample size was not as large as the 76,000, we had a pretty robust sample size. And these are the main signals that come out of this: BEN1, which you know, CD2AP, which was already mentioned today, EPHA1, clusterin, MS4, PCOM, and ABCA7. The ABCA7 consistently shows up. This was an interesting family because it had an autosomal dominant pattern for an EPHA1 mutation. This shows the expression now in those individuals, both from this family and from our case control cohort, looks at the expression, so it seems to be an increased expression associated with Alzheimer's disease. And in the first fly model, which is not published yet, it has a hyperexcitability phenotype in addition to a learning problem. I don't have the movie to show you, but it's you have to take my word for it that it's a very interesting but yet unpublished phenotype. ABCA7, first by the group in Iceland, defined several of these mutations in ABCA7. Let me show you a panel of where they are. So if you look at where the mutations are, these are the mutations in the Caucasian population. This is, again, in that region I showed you earlier for ABCA7. This is a set of African Americans that have a mutation in this region. These are the ones from the Hispanics. These are mostly Dominicans. And in about 2 African American families and 2 Dominican families, we have a deletion accounting for the association in this wide region of ABCA7. So this gives us a real tool to work with when we go to look at the functional aspects of ABCA7. What about whole exome and whole genome sequencing? Well, the first one that actually took advantage of it was the association by John Hardy and several others to identify TRIM2 as a risk factor. They used the NIA load families, and they used other families that had been acquired to make this identification. But a more recent study includes rare coding variants, and this is now using all of the cohorts in the Alzheimer's disease sequencing project, the Alzheimer's disease Genome Consortium. And these 3 coding variants, these 3 genes with coding variants are novel including TREM2, but they all seem to work in this microglial immune-related protein-protein interaction network, suggesting, again, another aspect of immune-related disease. And if you look at the map, this is just, if you're interested in how we use these cohorts, put them all together in a discovery set, then use replication to be certain of the mutation and get it down to this series of genes. What's important in these network cartoons is to pick out the ones that are not only associated, but pick out other ones. If you remember, CD33 sort of dropped out of the GWAS loci after after the sample sizes got larger, but it's clear that it's playing an important role, and it may be an activator in this complex. The jury is still out as to whether CD33 is an important aspect, but we think it probably is. We also, because of the blurring of lines between early and late onset disease, we've actually been collecting early onset families also that don't have presenilin 1 and 2 mutations or don't look like they do, but of course we find them. And another interesting factor has been that mutations in SOR1 are now showing up in early onset autosomal dominant looking families. And in fact, our colleagues in Europe have now suggested that pathogenic SOR1 variants should be considered for AD risk assessments along with ApoE, presenilin 1 and 2, and APP. I'm going to introduce a new study that we just finished. This was a whole exome study of a large number of Dominican families, and we uncovered, this is one of the 12 families that carry this mutation. These are mutations in a gene called SRCAP. It's a SNF-related protein that is involved. It's an activator protein for the CREB binding protein. And we had 12 mutations in the gene, and they were in families that looked something like this family. The red are the carriers that are affected, and here's a married-in person who also is affected but is not a carrier. Interestingly, this gene is associated with a congenital syndrome called floating harbor syndrome, in which the child are born with some developmental problems. They don't speak for the first couple of years of life, and they have some skeletal abnormalities. And that's due to truncating mutations in exon 34, where we do see some missense mutations, but nothing that disturbs the holoprotein. These mutations also, though, have an effect on the pathology. So as the expression goes down due to these mutations, as expression goes up, rather, cognitive performance deteriorates and pathology goes up. This is data from the Ross MAP study that we, in our collaborators. So SRCAP can cause mutations in the floating harbor syndrome. It's an activator of the CREB binding protein. It regulates the CREB pathway, facilitating expression, and mutations are associated with both the clinical and pathological features of Alzheimer's disease. So the Alzheimer's sequencing project had initially 159 families. These are multiplex families. Most of them are from the Caribbean Hispanic cohort that I direct, and also from the NIA load families, and 10 from Erasmus. We've done whole genome sequencing in these families. The work is completed. The manuscripts are under review. The data is all available, so if you want to get access to the data or to the families, you can simply do that. And we also just finished whole exome sequencing in 5,000 unrelated cases and controls. And then we chose 1,000 families that were from 1,000 individuals from families that were cases. We call them enriched cases because we think this may be a good cohort to look at. So this is just unpublished data. This is the first look at the ADSP family data. This is from the first half of the families. We have a couple of signals that are very interesting. And if you look at the genes, the most important ones, I think, are PLD1 and SPAG17, both of which in subsequent analyses have shown to be getting stronger with multiple variants being identified, and that paper's under review and should be published quite soon. In the case control design, especially in the enriched case control design, our old friend TRIM2 shows up again. So does cancel1. This had been previously identified as a modifier of APO in individuals who are APOE negative, missense mutation, but probably the newest one is this homeobox mutation, which is quite robust and was present in a large number of individuals. Again, the functional significance of these genes needs to be elucidated. For those of you who are interested in getting information, either the summary statistics or you actually want to get the raw data yourself, this is the place to do it. It's the Alzheimer's Disease Sequencing Project storage site. So everybody, we can facilitate ADSP data. If you have your own data and you want to join the program, we can help you harmonize it. We do the bioinformatics to get it up to par with the other data, and then you become a partner and you can access the data pretty easily. We encourage anybody who wants to work with this data to contact us, and more information is on how to do everything is on the ADSP website. It looks something like this. You have to create a password. You have to try to remember it in the future. That's not a test, but it helps. But it's very accessible. So right now, as I said, we've finished with the first 159 families. Work's completed. What we're doing in the family analysis is that we've just added 112 individuals from 46 of the existing families and 177 individuals across 147 new families, and added some controls from the Hispanics since it's an unusual population. We've just finished phase 2 where we added another almost 300 families here. These we expect to get in the following year, bringing us to a total of 572 families, roughly 3,000 individuals who have whole genome data. And again, it's publicly available data, so if you want to analyze the data, you can gain access to it. We will be analyzing it ourselves. For the case control, a large study has just begun. This is a collaboration between Peggy Vance, Badri Vardazian in my unit, and myself. We are now about to start sequencing another 10,000 individuals. This is a cohort from Mexico, a couple of Puerto Rican cohorts, a couple of Dominican cohorts, African Americans, and then a big autopsy series that we've been able to put together. So taking a step back from all the information I've provided you with, it turns out that these risk variants are starting to cluster into certain patterns. And even today, we heard a little bit more evidence about BIN1 and CD2AP as being part of this recycling process. The immune response, the numbers keep growing there as well. Some of these genes have multiple effects. You see ApoE everywhere because they keep changing its important function, but it's probably true. And cholesterol and lipid metabolism is over on this side. EPHA1 seems to be important in synaptic maintenance, but this map will probably shorten, and once we get most of the variants identified, we then will be able to mock up some potential pathways to exploit for therapeutic purposes. One caveat on all of this: we have all been thinking of Alzheimer's disease as plaques and tangles, and unfortunately, in late onset disease, only about 20 to 30% of patients have only neurofibrillary tangles and amyloid plaques. Many of them, as many as 30%, have Lewy bodies or Lewy neurites, and many more have cerebral vascular disease. We sort of ignore these two units where it's important to note that these are probably important. So as our numbers pick up, we're going to have to use markers for Lewy bodies to try to dissect out the AD Lewy body overlap. And right now we're using MRI basically to understand the overlap with cerebral vascular disease. That will have to be taken into account. So the implications are genetic variation either directly or indirectly underlies the cause of Alzheimer's disease. With few exceptions, multiple rare and ultra-rare variants underlie most of the genetic loci that we see. Signs and symptoms may be similar, and pathological and etiological differences likely result from genetic variation and environmental interaction. I think the only way we're going to have success developing treatments is if we map out all of the genetic influences in the disease so that we can begin the molecular approach to understand how to develop targets to treat these things. And I guess treatment in the clinic will be based on your genetic profile. These are my collaborators. This is Jerry Schellenberg, Peggy Vance, Lee San Wang, Jonathan Haynes, Lindsay Farr, Tatiana Faroud. This is my lab back home. And this is my 2 colleagues in the Dominican Republic. And this is my funding. Thank you very much.
Any questions from the audience? One right in the middle of the... Can you go up with the microphone?
Hi, my name is Hanne Stensula, and I'm a postdoctoral fellow here at the Champollion Center. I had a question regarding the methodology that you use when you're looking for these genes that cause the sensitivity to developing diseases that we know have multiple factors. Instead of looking at just a single gene that sticks out, what if there are multiple genes that alone does not imply any risk, but in a certain type of combination, when that appears, implies this risk? Do you do any analysis that shows out such combinations?
Yes, let me just rephrase your question. You're asking, how do we account for 30 genes for a single phenotype? So there's 2 ways we do that. One is, in some of these analyses, we try to determine who had, when we do whole exome or whole genome, we map out everyone who has what we think is the abnormal variant. And for those, we almost always add additional genes like ApoE, or we condition on these others. In families, it's a little bit easier because you do have to correct for ApoE, but in families, generally, the cause of the disease in the family tends to be in one set of genes. Now, the problem with families also is that if you have a family that has a mutation that you think is associated with Alzheimer's disease, there could be some other characteristic that same mutation is related to and has nothing to do with Alzheimer's disease. So we hope to see additional families or other case control or other cases that are not related to the family that carry the mutation that are also affected. But this is an age-dependent disease, so sometimes people carry the gene, they're not quite affected, they may have some cognitive impairment, or they may be perfectly normal and they're young. So all of the things you're worried about, we worry about too, and we try to address those as best we can. There are the polygenic risk scores that I mentioned, but that's sort of at the locus level, it's still not at the variant level.
Other questions?
Yeah, I would actually add one. So, how do you think about gene-environment interactions in case of humans? Obviously, gene-lifestyle environment interactions, right? Because these could really also give important insight in terms of personalized prevention of the disease.
Well, I spent the first part of my career looking at environmental factors, and again, replication was always hard there. And I think the direction of causality is always in question there. So techniques like Mendelian randomization can help you with some of these frequently associated risk factors, such as education or heart disease or things like that. And there have been just now, there are starting to be a few papers that use that approach. Again, they're using GWAS loci, they're not using variants, but eventually we'll have enough variants to actually be able to do that. Thank you.
Thank you very much.
Over there.